Yeah, coming to next chapter, heme metabolism. So we know that hemoglobin it is a conjugated protein having protein part plus non-protein part. The protein part is globin and the non-protein part is heme. So here heme, the non-protein part of hemoglobin, is nothing but it is a porphyrin ring with iron atom. So what is porphyrin ring? So porphyrin ring is nothing but it has 4 pyrrole rings, and 4 pyrrole rings they condense to form a porphyrin ring. So that's how heme is formed. So the structure of heme is it is a porphyrin ring with iron atom, and porphyrin rings are formed by condensation of 4 pyrrole rings. So this heme synthesis mainly occurs in the bone marrow. So it accounts for 85% of heme synthesis, and the remaining occurs in the liver. Right? And when you see location of this heme synthesis, location of the pathway, it occurs in cytosol and mitochondria. So it starts in the mitochondria, and remaining reactions they occur in the mitochondria, and the last reaction occurs again in the mitochondria. So first and last reactions they occur in mitochondria, remaining reactions in the cytosol portion. And coming to its synthesis, and for the synthesis of heme we require 2 simple compounds like succinyl-CoA and glycine. So succinyl-CoA is obtained from the source for succinyl-CoA is citric acid cycle, and glycine is the simplest amino acid. So glycine and succinyl-CoA they react, and the condensation of these two is catalyzed by the enzyme called as allosynthase. So by the action of allosynthase, condensation of glycine succinyl-CoA occurs, and it leads to synthesis of ALA. So ALA is delta amino levulinic acid. So that allosynthase, which is catalyzing the reaction, this reaction is the key enzyme, rate limiting enzyme of the heme synthesis. So this allosynthase activity is increased by barbiturates, a type of drugs, and the activity is decreased by hemine or hematine, and so these are the positive and negative inhibitors. So it is increased by the activity of allosynthase is increased by barbiturates and decreased by hematine. And what happens to the ALA? So that ALA is converted into porphobilinogen by the action of enzyme called as ALA dehydratase. So 2 molecules of ALA they react, and dehydration occurs here, and that enzyme is ALA dehydratase to form porphobilinogen. So there porphobilinogens again by the action of enzyme called as porphobilinogen deaminase, and by the action of another enzyme called as uroporphyrinogen 3 synthase. So porphobilinogen deaminase acts on porphobilinogen and converts that into hydroxyl methylbilane. So that hydroxyl methylbilane now by the action of enzyme called as uroporphyrinogen 3 co-synthase is now converted into uroporphyrinogen 3. So that uroporphyrinogen 3 by the action of enzyme called as uroporphyrinogen 3 decarboxylase is converted into coproporphyrinogen 3. So what happens to that coproporphyrinogen 3? It is by the action of enzyme called as coproporphyrinogen oxidase is now converted into protoporphyrinogen 9. So by the action of enzyme, coproporphyrinogen oxidase is converted into protoporphyrinogen 9. That protoporphyrinogen 9 further undergoes oxidation and is now converted into protoporphyrin 9. So that protoporphyrin 9 by the action of enzyme called as heme synthase, ferrochelatase. So that ferrochelatase or chelatase that incorporates iron into the protoporphyrin 9 so that it is converted into heme. So for the synthesis of heme, in the final step we require ferrochelatase. That ferrochelatase incorporates iron into protoporphyrin 9. So that is the final step that occurs in the mitochondria. So in this way heme is synthesized from the simple precursors like glycine and succinyl-CoA. So here the key enzyme is allosynthase, and so in this picture you can see the synthesis of heme starts in the mitochondria and ends in the mitochondria, and the remaining reactions they occur in the cytosol portion. Right? So the key enzyme of heme synthesis is now present in the mitochondria. Coming to regulation, how this heme synthesis is regulated. So as we discussed that whenever there is increased synthesis of heme, sufficient heme is synthesized, that heme inhibits the key enzyme of heme synthesis, that is allosynthase. So this type of regulation we call it as feedback regulation. So by feedback inhibition, heme synthesis is regulated. And the other way in which heme synthesis is regulated is by allosteric inhibition. Right? When you take hematin, whenever there is heme in excess is there, that heme is converted into Fe3 plus form, ferric form, and that form of iron we call it as hematin. So that hematin binds with the key enzyme again allosynthase and inhibits the activity of allosynthase, and this mechanism we call it as allosteric inhibition. And the third mechanism is compartmentalization. So here we discussed that some reactions they occur in the mitochondria and some occur in the cytosol. So here the reactions, the key enzyme which catalyzes the heme synthesis is present in the mitochondria. So in this way, by compartmentalization of enzymes, we can regulate the heme synthesis. And the other mechanism is drugs. So the key enzyme of heme synthesis, again allosynthase, it is activated by drugs like barbiturates. So the activity of allosynthase is increased when a person takes these drugs, barbiturates. Right? So this is about the regulation of heme synthesis. Now coming to the inborn errors concerned with the synthesis of heme. So whenever there is any defect in the enzymes which is concerned with the heme synthesis, it leads to a condition called as porphyrias. So porphyrias are the inborn errors of heme synthesis. So whenever any enzyme which is concerned with the heme synthesis defective or deficiency is there, then it leads to porphyrias. So we have different types of porphyrias based on the block in the heme synthesis. So based on the enzyme which is defective or deficient, we have different types of porphyrias. So when you take the most important one, acute intermittent porphyria, so that is because of deficiency of enzyme called as porphobilinogen deaminase or uroporphyrinogen 1 synthase. So deficiency of this enzyme leads to a type of porphyria, we call it as acute intermittent porphyria. So why it is named as acute intermittent porphyria? Here the symptoms are acute and intermittent. So symptoms are seen intermittent, they are not continuous. Intermittent symptoms are seen, so that's why it is called as intermittent acute porphyria. So acute intermittent porphyria. So when you see the important features of this porphyria, in this porphyria we can see dermatological features, right? So another one is neurological manifestations, right? So neurological manifestations like agitation, confusion and psychic nature is seen in case of acute intermittent porphyria. And apart from that, you can see abdominal pain. So that is another important features. So no photosensitivity, no dermatological manifestations, only abdominal pain and neurological manifestations are seen in case of acute intermittent porphyria. And coming to the other type of porphyria, that is congenital erythropoietic porphyria, and that is because of the deficiency of enzyme uroporphyrinogen 3 co-synthase. So deficiency of this enzyme leads to a condition called as congenital erythropoietic porphyria. So this is seen in the mainly seen in the erythropoietic cells. So that's why it is called as erythropoietic porphyria. So if the defect in the hepatocytes, we call it as hepatic porphyria. If the defect is in erythrocytes, we call it as erythropoietic porphyria. So here it is a defect is present in the erythropoietic cells. So it is called as erythropoietic porphyria, and that is congenital erythropoietic porphyria. So we know the enzyme deficiency is uroporphyrinogen 3 co-synthase. So what are the important features of this congenital erythropoietic porphyria? So here you can see mainly dermatological features like so because of the accumulation of porphyrins, so those porphyrins they get excited when light falls on them. So when they are exposed to light, they get excited, and that excitation leads to itching, and that itching leads to dermatitis and scary nature. Scars will be seen on the skin. So these dermatological and skin manifestations are seen mainly in case of congenital erythropoietic porphyria. Apart from that, there is hemolysis. So these are the important features of congenital erythropoietic porphyria. And now coming to the other important feature, another important porphyria, that is Porphyria cutanea tarda. So that is because of the deficiency of enzyme called as uroporphyrinogen decarboxylase. So that uroporphyrinogen decarboxylase deficiency leads to this porphyria, and the name itself indicates that there are skin manifestations, Porphyria cutanea tarda. So skin manifestations are seen because of the porphyrins which are accumulated in the blood. So there is accumulation of porphyrins. So that is because of the deficiency of enzyme uroporphyrinogen decarboxylase. So when uroporphyrin decarboxylase is defective, it leads to accumulation of uroporphyrinogen 1 type. So that one type of the porphyrins are accumulated, and those porphyrins they get excited when light falls on them. So that excitation is the result of dermatological skin manifestations, and that leads to photosensitivity. So that's why it is called as Porphyria cutanea tarda. And apart from that, we have some more porphyrias like hereditary coproporphyria, Porphyria variegate porphyria, and another one is Porphyria hereditary protoporphyria. So these are the other types of porphyrias, and that is because of defective enzymes in the heme synthesis. So any enzyme which is concerned with the heme synthesis, whenever it is defective, it leads to accumulation of corresponding porphyrins. So together we call all these conditions as porphyrias. And now you have seen that how heme is synthesized. So heme is synthesized from the simple compounds glycine and succinyl-CoA, and after its heme synthesis, so it binds with the hemoglobin, it binds with the globin and is converted into hemoglobin. So that hemoglobin in RBCs, when it is lysed, so lifespan of RBCs is 120 days. After that, lysis of RBCs occurs, and once it occurs, hemoglobin is released. From hemoglobin, heme is separated. That fate of heme is we have to conserve that iron. So heme is subject to degradation. So in its degradation, first heme, by the action of enzyme called as biliverdin, heme oxygenase is converted to biliverdin. And now biliverdin, by the action of enzyme called as biliverdin reductase, is converted to bilirubin. So these two enzymes are responsible for the degradation of heme. So those two are heme oxygenase and biliverdin reductase. So those two enzymes convert heme into bilirubin. So there the degradation of heme occurs in the form of bilirubin. So that bilirubin which is there formed now undergoes conjugation with glucuronic acid, and it is excreted from the body in the form of bilirubin diglucuronide. Right. So that glucuronic acid, when it conjugates with bilirubin, it is acted upon by the enzymes. It is converted into urobilinogen in the intestine, so that is excreted from the body. So basically, heme catabolism leads to the formation of bilirubin by the action of two enzymes, heme oxygenase and biliverdin reductase. So till now we have discussed about how heme is synthesized and where it is synthesized. So what are the inborn errors of heme metabolism, why you call them as porphyrias, and how heme is catabolized, how it is degraded in our body. Thank you.